So we will start off with a woman whose roots lie in the Department of Immunology, the Institute of Cell Biology here in Tübingen. Almost 10 years after she started, she now has her own research group within this department where she investigates the immunopeptidome in various cancers. Furthermore, besides the great number of high-end publication, awards, patents, and other accomplishments, which show that she's an amazing researcher, she's also incredibly kind and encourages you to pursue a scientific career with her enthusiasm. So without further ado, I would like to introduce you to Privatdocent Medical Doctor Juliane Walz.
Is everyone able to see the presentation?
Yes.
Great. Okay, perfect. Then I would like to thank you for inviting me to this workshop. Today, I would like to give you a short overview on the work of our research group that is focusing on the development of peptide-based immunotherapies. So with the development and the clinical success of immune checkpoint inhibitors, we now have for the first time the possibility to take the breaks from anti-cancer T-cell responses. However, so far this treatment is only available and effective in a small proportion of patients and cancer entities. To further increase the number of patients that profit from T-cell-based immunotherapy, one option is to use the targets of anti-cancer T-cell responses, which are represented by HLA-presented tumor-associated peptides on the cell surface of tumor cells that can be used to develop more specific anti-cancer immunotherapies. This includes, amongst the development of TCR-engineered approaches, as well as HLA peptide targeting antibodies, especially so-called peptide-based vaccines, which is the focus of the Department of Immunology for many, many years now. So how is the underlying mechanism of such a peptide-based vaccine? We have today the ability to produce tumor-associated peptides synthetically as a drug component and apply it to the patient's skin, where they are uptaken by antigen-presenting cells, and then prime T cells in the lymph nodes. And in the best case, there's then a proliferation of peptide-specific T cells that are able to detect the respective tumor-associated peptide on the cell surface of tumor cells and could eliminate the tumor cells. So the aim of my research group was to develop such peptide-based vaccines for, in the beginning, hematologic malignancies. We are now spreading out also for solid tumors or viral disease, as I will show you in the end of this talk. So before we are able to develop a peptide vaccine, we have to think about the requirements for a good peptide vaccine. The first and I think most important requirement is the selection of optimal antigen targets. The other thing we have to think about is how we could combine a peptide vaccine in the landscape of cancer treatment, and how we could select suitable combinatorial therapies. And the third thing is we need to have strong adjuvants to induce strong and especially long-lasting T-cell responses. So to start with the first requirement, the selection of tumor antigens. So what are the requirements for a perfect tumor antigen? The first thing, it has to be immunogenic. That means it has to be able to induce antigen-specific T-cell responses. The second thing is it has to be naturally presented on tumor cells so that the T cells could have a target and could be clinically effective. And furthermore, these targets should not be presented on any benign cells to reduce side effects due to off-target effects. So what kind of tumor antigens do we know? There are two big categories that are described. So the first are the non-mutated antigens, which are on the one hand so-called tissue-specific or differentiation antigens that are presented and are specific on a specific type of tissue, like for example, prostate tissue, where we have antigens that are presented exclusively on prostate as well as on prostate cancer, but on no other tissue of the body. The second group are the so-called overexpressed self-peptides. These peptides arise in a tumor cell due to differential gene expression or differential translation or processing processes, and are therefore tumor-specific. The second big group are the so-called mutated antigens or neoepitopes that arise from tumor-specific mutations in a cancer cell. So in recent years, we have also focused on a third group of potential tumor-associated antigens, the so-called inducible or treatment-associated or stress-associated antigens. These are tumor antigens that are induced due to the treatment with a specific drug or due to a specific cell stress condition specific in a tumor cell. And I will give you a few examples during my talk for these inducible antigens. So coming back to the neoepitopes, these antigens have been discussed as the preferential tumor antigens in recent years, as they have been described as the main specificities of T-cell responses that are induced or mediated by immune checkpoint inhibitors. Therefore, several peptide vaccine trials have been developed to analyze these neoepitopes in a clinical setting, as here shown for a small study evaluating a personalized neoepitope vaccine in melanoma patients. However, it seems as if these mutation-derived neoepitopes are mainly specific for cancers with high mutational burden, for example, melanoma and lung cell squamous carcinoma, as these entities are the entities with the highest response rate to immune checkpoint inhibitors. Furthermore, the design of these personalized neoepitope-based vaccines are so far very complex, expensive, and also quite ineffective. For example, this trial I showed you before vaccinated in total 97 HLA class 1 peptides, and only for 15 of these peptides a CD8 T-cell response could be detected. This might be due to the fact that the prediction of these neoepitopes based on genome data does not guarantee the natural presentation of these mutations on the tumor cell. This was shown exemplarily in a work analyzing colon cell carcinoma cell line, where out of the about 30,000 exome variations, only 3 naturally presented and immunogenic neoepitopes could be validated in the end. So this calls for a method that enable us to really prove the natural presentation of tumor-associated antigens that are applied in peptide vaccines. And this method was developed by the Department of Immunology many years ago and was optimized in recent years, which allows us to directly isolate HLA-presented peptides from the cell surface of a tumor cell by affinity purification, and followed by the analysis of the isolated peptides by mass spectrometry. This allows us to characterize the whole amount of HLA-presented peptides on a tumor cell, the so-called HLA ligandome or immunopeptidome. This approach enabled us in recent years to analyze multiple solid tumors and hematological malignancies, as well as samples, organs, and tissues from healthy donors. and to develop the Tübingen HLA ligandome database that includes so far several million different HLA class 1 and class 2 peptides from a vast amount of different tumors as well as benign tissues. To now identify within this database the antigens that are really tumor associated and could be used for broadly applicable peptide vaccines, we are using this comparative ligandome analysis, as you could see here in this modified waterfall plot. So each black line represents antigens that are identified on malignant cells, and each gray line represents antigens that are identified on benign cells. And as expected, most of the HLA-presented peptides we are identifying are represented both on malignant and benign tissue and are not tumor-associated. But if you have a look on the left side of this graph, there is a group of tumor-associated antigens that are identified with a high frequency exclusive in malignant tissue. And we define these antigens as ligandome-derived tumor-associated antigens, which were further characterized as potential vaccine candidates. So one question was if these ligandome-derived tumor-associated antigens are the same antigens that were already described in literature as tumor-associated. Here I would like to show you an example of our project in chronic myeloid leukemia, where several tumor-associated antigens are described in literature. And what we could show is that for nearly all of these antigens, we are able to identify HLA-presented peptides. However, most of these antigens are not CML exclusive on HLA ligandome level, but are also identified on benign hematological samples like bone marrow or blood. Only here two HLA presented, for two antigens, HLA presented peptides were presented exclusively, however, in a really low frequency on CML samples. So this is the case if we have a look on the protein level. If we go on the peptide level, as shown here, for example, for the protein FLT3, which is overexpressed and in some cases also mutated in acute myeloid leukemia, we could see that we identify peptides from FLT3 in benign hematological tissues, shown in blue, as well as in hematological malignancies. But what we also could see is that there are areas of the protein that are only presented in malignant tissues, in this case AML, which might represent really interesting tumor-associated peptides, even if the whole protein is not tumor exclusive. The approval of mass spectrometry sensitivity and detection methods has enabled us in recent years to not only identify tumor-associated peptides from bulk tumor tissue, but also enable us to go in more detail and to characterize immunopeptidome of specific cell groups. In this case, leukemia progenitor, leukemia stem cells, which we were able to isolate and enrich, and we could also show that enriched leukemia progenitor cells were able to induce de novo leukemia in a not sick mouse model. And we characterized the immunopeptidome of these leukemia progenitor cells in comparison to a benign data set as well as to AML bulk. And we were able to detect a small amount of leukemia progenitor cell exclusive antigens. And most important, we could also detect a high frequent amount of peptide that is enriched on the leukemia progenitor cells as well as on the bulk blast cells, which are highly interesting candidates for the treatment of AML, especially minimal residual disease in leukemia. In the next step, we wanted to know if the antigens that we identify across different hematological malignancies could be used for a so-called pan-leukemia vaccine that could be applied for all hematological malignancies. Therefore, we did this cluster analysis of all HLA ligandosomes from different hematological malignancies, including AML, CML, CLL, and multiple myeloma. And what we could show is that there is a clustering of lineage and tumor entities of the different ligandomes, which means, on the other hand, there are no pan-leukemia associated antigens that are high frequently presented on all different malignancies, which means for the future design of peptide vaccines, we have to focus on each single entity, and even more, we have to think about focusing on the individual patient and on personalized peptide composition, which will be, I think, the focus on Marco's talk later. So we also wanted to know if the approach of direct mass spectrometry immunopeptidomics is able to identify mutated antigens that are naturally presented on tumor cells. In recent years, the group of Bassani-Sternberg could show that for malignant melanoma, which has the highest mutation frequency amongst the tumors, they were able to detect a few naturally presented neoepitopes in this high mutational burden tumor. So we wanted to know if this is also possible in the hematological malignancies which show a very low mutational burden, but show a very distinct mutation pattern. So we focused on our AML cohort, where we have mutations that are presented in up to 30% of all AML samples, which is on the one hand the NPM1 mutation. So we had 22 NPM1 AML samples, and we were able to identify in 4 of these samples NPM1 mutated HLA ligands for A3 and A11. And we were further able to identify on HLA class 2 one mutated peptide from ADH2. So we could say we are able to identify naturally presented mutation-derived peptides also in low mutational burden hematological malignancies, but we find these peptides in a very low frequency, which might be due to HLA allotype restriction in this case, so that these mutated neoepitopes could only be used for a personalized design of vaccine and could not be broadly applicable. So we further wanted to know why we are not able, for example, to identify mutated peptides from the FLT3 mutation, which is in case the most common mutation in AML. And therefore we again screened the whole FLT3 protein, as I showed you before, for peptides that were identified across the whole amino acid sequence. And of interest is that the regions where the ITD and the TKD mutation of FLT3 are located did not present any peptides, not on AML, but also not on benign tissue, which might explain that this part of the protein is not processed and presented on HLA, which might be one explanation why we could not identify any mutated FLT3 peptide in our data set. So the next step of validation of our tumor-associated peptides is the approval of immunogenicity. This is performed on the one hand by in vitro priming of naive T cells by an artificial antigen presenting cell approach, which enable us to induce peptide-specific T cells targeting our peptides, which are multifunctional and are also able to lyse peptide-loaded cells, as well as cell lines that present these peptides. Furthermore, we are proving the immunogenicity of these peptides by screening our patient cohorts for the presence of pre-existing memory T-cell responses against these peptides. Here I would like to show you the example of our CLL cohort, where we screened these patients for pre-existing peptide response and could identify in a set of patients pre-existing T-cell responses targeting our CLL-associated antigens, while we never found these immune responses in healthy individuals, proving the specificity of this T-cell reactivity. We further wanted to know if there is any pathophysiological relevance of these immune responses in the small set of patients that show these pre-existing immune responses. And what we could show for CLL patients that showed an immune response to more than one of our CLL-associated antigens had an improved overall survival. Which leads us to the next question. If we would be able to induce immune responses in the group of patients that showed no pre-existing T-cell responses to our peptides by peptide vaccination. So the development of clinical studies evaluating peptide vaccines is enabled by the Herstellungserlaubnis for individualized peptide vaccines and the GMP Center for Individualized Substances in Tübingen, which enables us to develop these peptide vaccines and produce them by our own. So this enabled us to develop and start the first patient individualized peptide vaccination study in CLL patients. This study includes CLL patients after first-line treatment of CLL, and the patients get their first-line treatment, which takes about 6 months. And during this time, we are producing an individual peptide vaccine. This is based on the patient individual HLA ligandome analysis, and to enable a fast and cost-saving individualized vaccine, we have set up a so-called warehouse concept where we pre-produce the most common CLL-associated antigens in this warehouse for several HLA allotypes, so that we then could select, based on these patient individual ligandome analysis, an individualized cocktail out of this warehouse. So far, we have included 24 patients in this study. 20 patients were already vaccinated and 16 patients have finished the study. We could approve the feasibility of this warehouse approach because we were able to identify for 94% of the patients at least one naturally presented warehouse peptide in the HLA ligandome of the CLL samples. The vaccine is quite well tolerated. We have seen no systemic side effects so far. We see only this local immune reaction, which lasts for about 2 to 3 weeks. And we have analyzed the first 12 patients and could detect peptide-specific immune responses for 9 of these patients, as exemplarily shown in the immune monitoring for 1 patient after 10 months that showed an immune response to 1 of the HLA class II peptides. So to further develop such peptide vaccines that take place not only after the end of standard treatment, but could also be combined with standard treatment and are even able to induce more and stronger immune responses. We focused in recent years also on the development of novel combinatorial therapies, which might be the combination of peptides with immune checkpoint inhibitors, but also with other standard treatments. One further option to improve peptide vaccination might be the combination of non-mutated and mutated peptides, which was done in the GAPVAX study, which used a very complex approach to combine unmutated glioblastoma-associated peptides as well as mutated glioblastoma peptides and applied them to glioblastoma patients. And what this study could show was that there were immune responses to mutated and non-mutated peptides. And while mutated peptides mainly induced CD4 T cells, non-mutated peptides mainly induced CD8 T cells. So this might be really interesting to combine these peptides in a vaccine. To select the optimal combination therapies for peptide vaccination, it's on the one hand of high importance that these combination therapies do not hamper T-cell response or even increase T-cell response, but it's also of great importance to know what these peptides do with what these combinatorial treatments do with the immunopeptidome. Therefore, we have developed a treatment model that enables us to monitor the effects of the immunopeptidome longitudinal and quantitatively in patient samples treated in vitro as well as in vivo, as well as in cell lines treated with specific substances. So this approach enables us to longitudinally monitor the effect of specific drugs on single tumor-associated peptides of interest, as well as on the whole immunopeptidome. And it enabled us to really show that a drug mode of action is directly mirrored in the immunopeptidome. And most important, as I told you in the beginning of the talk, it enabled us to detect so-called treatment-associated peptides, which means these are peptides that are induced during a specific cancer drug treatment and are never identified on any untreated malignant tissue as well as on any benign tissue. To give you some examples of this work area, I would like to show you our work on the effect of the proteasome inhibitor carfilzomib on the ligandome of multiple myeloma. So proteasome inhibitors are approved treatments for multiple myeloma, and carfilzomib is a proteasome inhibitor that specifically blocks the beta ring of the proteasome. And we wanted to know how this affects the peptide presentation. And what we could show was that the treatment with the proteasome inhibitor carfilzomib leads to a selective downregulation of peptides with the allotypes A24 and A23, which we could show is due to a selective reduction of peptides with aromatic C termini, which is due to the specific block of carfilzomib of the beta subunit of the proteasome, where the chymotrypsin-like activity of the proteasome is located, which specifically cleaves after aromatic C termini. So this analysis is really important if we think about combining proteasome inhibitors with peptide vaccine, as we should avoid to include peptides with aromatic C termini in such a vaccine. In an ongoing project, we are further evaluating the influence of epigenetic drugs on the immunopeptidome of AML. Several other groups have shown that epigenetic drugs, like for example, demethylating drugs, can induce the gene expression of cancer testis antigens. Furthermore, a group in Heidelberg has shown that the combination of demethylating drug and HDAC inhibitors can induce the presentation of cryptic transcripts that have never been translated before. And we now wanted to know if these changes in genome and transcriptome are also present in the immunopeptidome and might result in novel treatment-induced targets. And what we could show so far is that treatment with of AML cell lines and primary AML samples with the hypomethylating agent decitabine is able to induce CTA-derived HLA ligands in the treated cells that might represent highly interesting vaccine candidates. And furthermore, together with the group of Heidelberg, we were able to show that the treatment of decitabine together with an HDAC inhibitor is able to induce so-called treatment-induced cryptic neoepitopes that are derived from these cryptic transcripts, and therefore might be also highly interesting candidates, as they are really neoepitopes not presented in any other tissue before. So the method of quantitative and longitudinal mapping of the immunopeptidome does not only allow to monitor the effect of specific drugs, but also the effect of cell stress conditions, like, for example, senescence. In this project, we are working together with Lars Zender, who is a mouse model that is and a specific component inducing senescence. And in this HCC mouse model, senescence is induced by the component CX5461. And what we have performed is MHC ligandome analysis of these HCC samples from that mouse model of senescent tumor cells, and compared them to the untreated tumor cells, as well as to benign liver cells that are treated with the senescence-inducing drug, as well as untreated benign liver and various benign other tissues from Black 6 mice. And we were able to identify high-frequent senescence-associated peptides that are currently evaluated in a vaccine in this mouse model to prove if this vaccine is able to eliminate the senescent tumor cells. So,
Juliane, can I shortly interrupt?
Yes.
In regard of time, is it possible that you can wrap up your presentation?
Oh, yes, for sure, for sure. I think it's only the last 2 slides now. I'm sorry.
Okay, great.
Sorry. So the third, and I think also very important part you need for a good peptide vaccine is to have a good adjuvant drug. And here Hans-Georg Ramense has developed a novel TLR1/2 ligand that is water soluble and therefore GMP producible. It's called XS15, and it has shown not seen before strong immune responses in healthy volunteers as well as first cancer patients. And we will evaluate this novel adjuvant in a follow-up trial of our CLL trial that was just approved yesterday by the regulatory authorities, and hopefully will start in the beginning of next year. We have already evaluated XS15 together with a checkpoint inhibitor in single tumor patients, and we could show that it is able to induce strong and long-lasting immune responses now up to 2 years. And we have seen first signs of clinical response in these patients. And I think I will skip the part on peptide-based immunotherapy of COVID-19 in detail. I just would like to tell that we also will start a first peptide-based COVID-19 vaccine study, hopefully tomorrow or on Monday next week. And yes, I would like to thank all people that are involved in these projects, and of course all the patients that participate in the trials. And I'm so sorry for taking too much time. Yeah, and I'm happy to hear your questions. Thank you.
Thank you very much, Juliane. Don't worry, the time schedule is still fine. Regarding the questions, I would like to take a look at the chat now. And I saw that Philip asked a question, but maybe it's nice to elaborate a bit more. So would it be possible to use the HLA peptidome to distinguish different tumors entities from each other, which is often a problem in pathology? Can you maybe elaborate on that a bit?
Yes, so in the project where we have compared the different hematological tumor-derived HLA ligands, we could show these clearly entity-specific clustering of the immunopeptidomes. However, I'm not sure if this clustering is more specific than the pathologist diagnostic to distinguish between the tissue. There is a clear clustering, and but I'm not sure if this would be better than the pathologist diagnostics. But it might be an addition to this diagnostic if there's any unclear diagnosis.
Thank you very much, Juliane. Maybe we can use the coffee break to start a discussion. And if anybody wants to have a word with Juliane, that you raise your hand. I don't know if that's okay with you too, Juliane?
Yes, no problem.
So Sven?
Yeah, I raised my hand and I'd have a question. So you talked about the interaction of like the HLA ligandome and potential treatment options together with other drugs like proteasomal inhibitors and epigenetic drugs, right? A couple years back, it was also used, I mean, small molecules, kinase inhibitors were also used in combination with immunotherapy. And as far as I remember, that was not successful, but I might be wrong on that. Well, is that still something that is investigated, or is that not used?
I think there have been approaches, like for example, the last phase 3 study of Emetics in renal cell carcinoma. They combined peptide vaccines with kinase inhibitors and were not successful to induce strong T-cell responses. I think the kinase inhibitors might have negative impact on T-cell responses. We've seen that for our CML project when we evaluate different tyrosine kinase inhibitors targeting the BCR-ABL transcript. However, these effects are specific for each individual kinase inhibitor. So I absolutely agree with you that before combining kinase inhibitors with peptide-based vaccine, this has to be taken into account. We have not analyzed so far the effect of tyrosine kinase inhibitors on the immunopeptidome. I think these inhibitors won't target the immunopeptidome so much, but it might be of, I think, utmost important to evaluate their effect on T-cell response.
Okay, thanks.
So we might have time for one person more. Is there somebody who would like to exchange some words with Juliane? And otherwise, I have one question maybe. So Juliane, you actually showed the setup for the patient's individual peptide cocktail. And based on the results, do you think that it will replace current cancer medicine, or that it will play a role in the current situation?
You mean the vaccination with a completely personalized vaccine?
Yes.
Yeah, so I think it would be the best way to develop these vaccines, to go on a personalized approach. However, there are several limitations, which is especially the GMP production, which is extremely time consuming and expensive, which might be one of the biggest limitations to provide such a completely personalized approach to a large set of patients. But Markus Löffler and Sebastian Hehn have set up a clinical study, the PepiVax study, which is evaluating such a personalized vaccine for several tumor patients to prove the feasibility of this approach. And I think the next steps then would be to find a way that production of such personalized vaccines will be more feasible and not limited by these strong GMP regulatories.
Thank you very much. I see that there's one question I would like to ask the organizers if that's still possible to ask them, or do I need to proceed to the next speaker?
I would say go ahead.
Well, that's good. So so there is a question from Leon. With the recent success of RNA vaccines, what are the advantages of using peptides for cancer immunotherapy?
So yeah, that's a really good question, and I think this will be a huge discussion in the future. So I think the biggest advantage is that the peptides that we use are really proven to be naturally presented on the tumor cell surface. Which is for the mRNA vaccine at the moment not the case. These are based on predicted neoepitopes. So I think this is our most important advantage, and I think also really important to get clinical effective results, that we prove this natural presentation.
Well, with this, I would like to thank Juliane for finding the time to give this informative talk, and also the Q&A session, which we just had.